3G, point gene mutations. This is where one or a small set of nucleotides are inserted, deleted, or changed in a gene. This one small change in DNA sequence can result in a change in the end shape, and therefore the function of the resultant protein. As with all mutations, this small point mutation may lead to a negative, positive, or neutral effect on the organism that carries the mutation. There are 4 types of point mutations, the first one being a silent mutation. Let's use this oversimplified template DNA sequence of GGG, CCC, TTT, and AAA. This will be transcribed into an mRNA sequence of CCC, GGG, AAA, and UUU. Using our genetic code, we can see that this mRNA sequence encodes for proline, glycine, lysine, and phenylalanine. Let's mutate the third G in the DNA sequence to an A, adenine. This will in turn lead to the mRNA sequence transcribed as uracil rather than cytosine, U instead of C. Fortunately for us, the codon CCU codes for the same amino acid, proline, as CCC did, which is a benefit of the redundancy of the genetic code. So the resultant protein will have an identical amino acid sequence as if there were no mutation at all: proline, glycine, lysine, and phenylalanine. The second type of mutation is called a missense mutation. If we return to our initial template DNA sequence of GGG, CCC, TTT, and AAA, we'll make a point mutation to the third thymine in TTT to adenine, TTA. The mRNA transcribed will have the third codon becoming AAU, which unfortunately codes not for lysine but rather asparagine. This may result in the protein working better or worse, or it may have no effect at all on the protein's final shape, and therefore its function. The third point mutation is called a nonsense mutation. Let's change the first T in TTT to an adenine, A. The DNA template now reads ATT, which results in the mRNA codon reading UAA, which codes for a stop codon. This would truncate the amino acid chain, likely causing the protein to function differently than before. However, if it's a positive, negative, or neutral mutation depends on how the changed protein affects the organism. The final mutation is a frameshift mutation. In this scenario, rather than changing a single nucleotide as we did with the first 3 mutations, this one will add or remove a single nucleotide, which essentially will change every single triplet codon downstream. In our example, we're going to insert a cytosine in between the first 2 guanines of the DNA sequence. By inserting the cytosine between the guanines, it causes a change in all of the codons downstream. The mRNA sequence transcribed becomes CGC, CGG, GAA, AUU. This codes for arginine, glutamic acid, and isoleucine, which is entirely different than the amino acid sequence of the original DNA sequence. It is quite obvious that frameshift mutation causes the most change in the amino acid sequence of the protein. However, if it's a positive, negative, or neutral mutation, entirely results on how the changed protein affects the organism.